Okay, so I wanted to go over again how eukaryotes control gene expression. And one of the first things that we learned about was the fact that transcription and translation occur in two completely different locations inside of a eukaryotic cell. So transcription, we know, is the formation of RNA from DNA that occurs in the nucleus. And then the RNA then goes to the cytoplasm for translation, where the polypeptide chain is assembled. And the next thing that we learned about were the introns and exons. And I forgot to show a video, which I'll show now, of transcription occurring. And you can actually see the, sorry, I'll back up a little bit. So the blue blob that you see there is the RNA polymerase. The red is obviously DNA, and then there's going to be two sections that are going to come off from the RNA polymerase. That's going to be the yellow exons and the green introns. Remember, exons are the ones that contain the codon sequence for the amino acid sequence in the protein, and the introns are nonsensicals. So you're going to see protein complexes and RNA splicing the exons together. So again, the green is going to have to be cut out, and the two yellow ends will be spliced together. You can see that involves a protein complex. So this is based on a computer model of the actual molecular structure of all of the proteins in the DNA. So you see one end of the intron was just cut, the other end, and now they've spliced together. Okay. Right, so introns, exons, we did that. Two different locations for transcription translation. All right, now, when transcription actually starts, there's another way to control gene expression or transcription translation together and the expression of a gene that, excuse me, that involves regulatory genes. And these genes are going to code for regulatory proteins called transcription factors. Now, transcription factors, you might think, I'd play this. You might think that transcription factors, again, this is going to be a computer model based on actual molecular shape. On the left-hand side here, you can see the DNA is wrapped around histones. That's called chromatin. And some of those, I don't know if you remember, I was said in class that some of the, turn that off because the noise bothers me. The narration from the video is distracting me. Some of the DNA is never uncoiled, so it's always in this, it's always in this wrapped up form. This form of DNA here cannot be transcribed because you need to have the double helix. So as we move in, you can see here's an unwrapped chain of DNA. This can actually be transcribed into RNA and then translated in the cytoplasm. Now, there's been some misconceptions about the function of RNA polymerase and transcription factors, which you see in blue here, that they just kind of randomly attach to different parts of DNA in the hope that it will randomly find the promoter region, when in fact what actually happens is what you're seeing now is that transcription factors are these proteins that are going to be basically continuously binding to a DNA molecule, or not binding to, but traveling down a DNA molecule until they find a promoter region, which you just saw there, which finds a region which it can bind to, and then it physically changes shape. Now, what happens after that is here. So it kind of looks a little bit like an operon, but what you have up at the top, I gotta exit because I need my pointer. What you have up at the top here is you got an enhancer region. And this is where the transcription factors actually bind to. The promoter, remember, is for the RNA polymerase. But RNA polymerase for eukaryotes cannot start transcription until you have these two transcription factors bind to the enhancer region. The enhancer region is going to be a bit further up on the DNA molecule from the promoter. So you can see that when the transcription factors actually bind, there's two binding sites. There's one on the enhancer and one kind of on the promoter / structural gene area. And once those two bind is kind of a signal. So when the transcription factor binds to the enhancer, it causes this loop in the DNA as kind of a signal to the RNA polymerase that it's okay to start transcribing. Okay, so there's a whole lot of stuff going on here, but basically I just want you to pay attention to the enhancer region. There's your transcription factors, because there's like, I think over 1,500 different types of transcription factors. And you see now how that all kind of comes together and combined. Now you've got these general transcription factors. It's a very complicated process, but basically you see, let's back up a little bit. So you see how there's an enhancer region here, this yellow part, and the orange part here, that's the promoter. So this is the two areas in yellow are the ones that are going to interact to, let's say, trigger RNA polymerase to start transcription. So play that again. A bunch of random protein, not random, but a bunch of proteins are going to participate in this process. So there's, see how that comes together like that, and then you've got transcription factors. Here's your RNA polymerase up here, so that's the one that binds to the promoter in the orange. But the general idea is you have a whole bunch of proteins and probably enzymes that are forming a complex together to bring this this whole section together as a signal for RNA polymerase to start transcription.